Hello, my juniors. Welcome to another biochemistry lecture series. It is your great science tutor Lorenzo, and today we're talking about what transamination reaction. So, if you are new to my channel, make sure to subscribe and turn on your post notification button so that when I drop any video, you'll be sure to be notified. And if you need me to make any video for you, or you have been studying and you do not understand some topics, just write it down in the comment section. When I go to it, I'll surely make the video for you. So let us jump right into the video. So now we'll be discussing what transamination reaction is. Now, you know, an amino acid have a carbon skeleton and an amino group. So now when the amino group is transferred to a keto acid to form a new amino acid and new keto acid, that's what transamination is. So simply, transamination process is the transfer of an amino group from an amino acid to a keto acid to form a new amino acid and a new keto acid. Simple. Now usually the keto acid is usually alpha ketoglutarate, because the enzyme transaminase or amino transferase uses alpha ketoglutarate as the acceptor. So alpha ketoglutarate accepts the amino group from the amino acid and turn to a new amino acid. Then the amino acid that gives out the amino group becomes a keto acid. Simple. So the enzyme is the transaminase or amino transferase. So the amino acid usually goes with the same name as the transaminase enzyme. So now, one thing I want to tell you now, that the coenzyme used in this case is what we call pyridoxal phosphate. So pyridoxal phosphate is the enzyme used. Pyridoxal phosphate is derived from pyridoxine, which is vitamin B6. So what does pyridoxal phosphate is? Now pyridoxal phosphate, which is the derivative of pyridoxine, which is vitamin B6, is covalently linked to the amino acid, which is of a specific lysine residue at the active side of the enzyme via a shift-based linkage, condensing both the amino group and the pyridoxal phosphate. So what the pyridoxal phosphate just receive the amino group, transferred, catalyzed by the amino transferase, then to form pyridoxine phosphate. So when you form pyridoxine phosphate, then what happens? It now combines with the keto acid to form the new amino acid. So simply, it just coordinates the transfer of the amino group before it is now connected to the keto acid to form the new amino acid and the new keto acid. So that is it. So since we discussed that transamination is the transfer of amino group from the amino acid to a keto acid to form a new amino acid and a new keto acid. This is catalyzed by a transaminase enzyme or an amino transferase with a coenzyme pyridoxal phosphate derived from vitamin B6. So simple. For the purpose of explaining transamination reaction, we are going to use two basic amino acids, which is alanine and aspartate. So alanine and aspartate are usually used in explanation of transamination reaction. So all amino acids can be used because they can form intermediate of TCA or glycolysis cycle, but with the exception of lysine and threonine. So lysine and threonine cannot be used as a transamination amino acid. So now using alanine now. So alanine has an amino group. So alanine amino group is transferred to a keto acid, which is alpha ketoglutarate. So if alanine amino group in alanine is transferred to alpha ketoglutarate, which is the acceptor. So what happened? By the enzyme alanine transaminase or alanine amino transferase, using pyridoxal phosphate as the transfer coenzyme. So what will it form? The alanine will form pyruvate, which is an intermediate of glycolysis, while the alpha ketoglutarate that received the amino acid will form glutamate. Very simple. So if the amino acid from alanine, if the amino group from alanine is transferred to alpha ketoglutarate, which is the acceptor of the amino group, it will form glutamate, while the alanine will form pyruvate. So the enzyme is alanine amino transferase or alanine transaminase. So that is very simple. So the next one we're using is aspartate. So aspartate does the same thing. So aspartate is the amino acid. Aspartate will transfer its amino group to alpha ketoglutarate, which is the acceptor. So aspartate will form what we call oxaloacetate, which is an intermediate of TCA, while alpha ketoglutarate will also form the glutamate, which is the acceptor of the nitrogen there. So the product of alpha ketoglutarate will be glutamate, while aspartate will form oxaloacetate. So what will be enzyme? The enzyme will be aspartate transaminase or aspartate amino transferase. Pyridoxal phosphate will also be the transfer coenzyme. So don't forget. So we are talking about alanine and we are talking about aspartate. So don't forget all these things. Don't forget all these examples, because ALT and AST are very important enzymes in the body, because elevation of them can cause different issues, which we'll be discussing. So clinically, elevation of aspartate transaminase and alanine transaminase is very important clinically because the elevation indicates a damage of a cell in the body, especially the cell that is producing it. So remember, even alanine transaminase and aspartate, ALT and AST, are usually produced in the liver. They are very rich in the liver. So damage of the liver due to cirrhosis or viral hepatitis or any form of liver injury, what will happen? There will be elevation of this coenzyme. But one thing you should know, that ALT is produced specifically in the liver, while what AST is sensitive to the liver. So all that, apart from the liver too, when you have myocardial infarction or any muscular disorder, these two enzymes will also be elevated. So clinically, when these two enzymes are elevated, what happens? They are indicating what? An issue in the body, structural issue in the body, which are, whether it's a muscle issue or it's just an organ damage. So don't forget it. I hope this all made sense, and with what I've explained about transamination and the reaction, which is the transfer of amino group from an amino acid to a keto acid to form a new amino acid and a two keto acid, and giving the two amino acid used, which is alanine and aspartate, to explain it. I hope it all made sense to you. Now, if you are just joining us for this first time, make sure to subscribe and turn on the post notification button so that when I drop any video, you'll be sure to be notified. So I keep saying it, that if you want a video to be made for you, write it down in the comment section. When I go to it, I will sure to make that video for you. So see you in the next class. Bye. On what oxidative what deamination, which is the deamination of the transaminated or amino acid. Bye bye.